All right. Okay, the next thing. We start moving on to the process of acetyl-CoA formation, Krebs cycle, and oxidative metabolism. Now, all three of those processes in eukaryotes occur in the mitochondria. So before we look at these processes, there will be questions on the test on this, by the way. Before we look at these processes, it's worth taking a good look at the mitochondria. What they are, how they're organized, what processes occur where, and a few other neat things about mitochondria. So let's take a look at these guys. Okay, mitochondria are small membrane-bound organelles in the cytoplasm of virtually all eukaryotic cells. There are a few exceptions, and we'll see that's a rather interesting case. They have not only one membrane, but they actually have two membranes. They're distinctly different, an outer and an inner membrane, and a fluid-filled compartment on the inside. Each cell is going to have anywhere from scores to thousands of mitochondria in them. Now, the classic textbook drawing of a mitochondria looks something like this, with the folded up inner membrane. Looks kind of like a sausage, a couple micrometers in size, micrometer or two in size. Turns out in real life, not all mitochondria are like that. They do vary quite a bit in shape. Some of them are quite elongated, some of them even branch. So this could be a mitochondria too. In fact, parts of one mitochondria could break off and merge with another mitochondria. That happens as well. So they're not just like the little fat sausages you see in the textbook diagrams. They actually are a lot more dynamic than that in real life. Okay. Now, let's take a good look at the mitochondria and the process that occur in here. So I'm going to draw an oversized mitochondria. We'll map out some of the processes. And I'll do the classic big fat sausage diagram. All right, big fat sausage. Okay. First of all, the mitochondria has an outer membrane. The main thing about the outer membrane is full of transport systems. Facilitate diffusion, active transport, what have you. To get all kinds of things in or out of the mitochondria. You need to get ADP in. You need to get ATP out. We would be in bad shape if the mitochondria made tons of ATP and just kept it all themselves, right? The rest of us wouldn't do so well. So we got to get things in. We got to get pyruvic acid in. We've got to get other things out. Even proteins have to be transported. Many proteins have to be transported into the mitochondria. And some proteins can be transported out of the mitochondria. So, the outer membrane is specialized for transport. Now, we're going to have an inner membrane. That inner membrane is folded up intensively into what's called cristae. So it's kind of folded up like a corner. I'm kind of drawing it like so, but in real life it's far more intensely folded than what I'm drawing here. Now, does anybody have an idea why we're folding this inner membrane up so much? We don't do that with the outer membrane. Why are we doing it? It's got to be a reason, right? Anybody? Not quite. Yeah. Close.
Surface area.
Surface area, yes. If we fold this up in real life, it looks more like an accordion. That means we pack an enormous amount of inner membrane in a small physical volume. And what that means is we have a lot of surface area of the inner membrane. And that surface area, if we want as much surface area as possible in the mitochondria, that must tell us something. That tells us that we must have a very important function for the inner membrane. And indeed, that is the case. And we'll see that in a minute. Now, there's this space between the two membranes. Now, this space is really very narrow. It's only about 20 nanometers across. It's really very narrow. It's 2 or 3 times as thick as the actual membrane itself. But that space is very important despite its small size. And we call this the intermembrane space. And as we're going to see here, what happens here is in the intermembrane space, we are going to actively transport H+ ions, enough that the concentration in there is almost 100-fold more than it is in the rest of the mitochondria or the cytoplasm. So we'll say H+ storage, and that's critically important for how the mitochondria work, as we'll see later. Okay. Now, in here, we have the fluid-filled compartment known as the matrix. That's a fluid-filled interior of the mitochondria. Now, let's see what happens where. First of all, let's look at the inner membrane. Turns out the inner membrane is where the process of oxidative metabolism occurs. Since that's a critical process, we want to pack as much of that as we possibly can into the small space of a mitochondria. That means we put as much membrane proteins in that mitochondria, in that inner membrane, as we can. As a matter of fact, if you look at the inner membrane of the mitochondria, in some ways it can be pictured more as a mass of proteins with each protein surrounded by a moat of lipid, rather than a typical pile of lipids with an occasional protein sticking in it. That membrane, by weight, is 80% protein, which is about what you get from a typical hamburger. So we have the process of oxidative metabolism occurs here. And that includes the electron transport chain enzymes, and this marvelous molecule that makes most of our ATP, appropriately called ATP synthetase. Okay, so we have the ATP synthesizing enzyme. All right. Now, let's see what happens in the matrix. There's lots of things go on in the matrix. The matrix is a fluid and enzyme-filled compartment. We go all the way into the inside of the mitochondria. And we see a number of important processes. We see the formation of acetyl-CoA. We have fatty acid for energy and amino acid metabolism, the enzymes that do that. And of course we have Krebs cycle occurring in the enzymes in the matrix of the mitochondria. Okay, so that's a lot of important metabolic stuff going on in there. But in the early 1970s, there was a discovery that shocked the biological world. Ordinarily, we think, especially in eukaryotic cells, the DNA is in that compartment we call the nucleus. But when they looked at mitochondria, and they also saw this in chloroplasts too, they realized that mitochondria and chloroplasts actually have their own DNA. That was fascinating. Nobody expected an organelle to have DNA in it. And as a matter of fact, the DNA was in the form of multiple copies of a circular molecule. Now, in the nucleus, our DNA in eukaryotes, they're linear pieces. In other words, they're not closed into a circle. You have a distinct in one end, and then the other end is all wrapped up with proteins. Here, the DNA is in the form of a closed loop. And like I say, most mitochondria have multiple copies of these rings of DNA. Now, what kinds of things have circular DNA? That's what you find in prokaryotes, in bacteria and in archaeans. Eukaryotes, except for mitochondria and chloroplasts, have their DNA in individual chromosomes, linear stretches of DNA with associated proteins. So this is very similar to what you see with bacterial type DNA. When you look at the organization and structure of it, it's also very similar to bacteria. You start sequencing things, it looks very similar to what you find in typical bacteria, and different than what you find in the nuclear DNA. Okay, well, mitochondria have DNA. Question is, does some of that DNA actually code for genes? And do some of those genes, so are we actually going to use mitochondrial DNA to make proteins, to write the instructions for proteins? And the answer is yes. They code for about 20 to 50, depends on the type of mitochondria the organism comes from, but they code for about 20 to 50 different proteins. Most of those proteins are very hydrophobic membrane proteins that are part of the electron transport chain or the ATP synthetase. So, they make their own proteins. Now, it turns out, to make a mitochondria, it's estimated you need between 800 and 1,000 separate proteins. The vast majority of proteins that make up a mitochondria are coded for by our nuclear DNA, and then the proteins are transported into the appropriate compartment. So mitochondria only make a tiny fraction of their proteins, but they do make some. Now, of course, if you're going to make proteins, that also means you have to have the protein synthesis factories, the ribosomes. And in the matrix, indeed, we do find ribosomes in there. Now, here again, the ribosomes in the mitochondria and chloroplasts are different than the ribosomes in the rest of the cell. The ones in the cell are larger, they have more components to them, they're more complex. They are sensitive to certain drugs and not to others. The ones in mitochondria and chloroplasts are smaller. In fact, they're very similar to the kinds of ribosomes you find in bacteria. About the same size, about the same composition, and things like that. And they are sensitive to the same kinds of antibiotics that kill off bacteria. Things like streptomycin and stuff like that. So those ribosomes are very much like prokaryotic ribosomes. Now, the other thing you need for protein synthesis is tRNAs, and indeed, they do have genes to make their own tRNAs. So you don't have to take tRNAs from the outside in, they make their own. So that is very interesting. It shocked the biological world when it was first discovered, except perhaps for one person who felt fairly vindicated. Because why do you have DNA in these organelles? Why do you make some of your own proteins? Well, way back in the 60s, a microbiologist just recently died named Lynn Margulis came up with a very controversial theory. She proposed that many of the organelles in modern-day eukaryotic cells were actually descended from prokaryotes. In effect, a eukaryotic cell is sort of like a town meeting of several different kinds of prokaryotes that merge together to make a much more complex cell. And each one of these prokaryotes, at least the descendants, became certain kinds of organelles. It was a very controversial idea, to say the least. Most people did not believe her. But looking especially at mitochondria and chloroplasts, she pointed out several lines of those. She pointed out that they have two membranes, both have two membranes. Same thing like with gram-negative bacteria. Those got two membranes too. She pointed out the lipids in mitochondria and chloroplasts are very similar to the lipids that you see in bacteria. You notice that mitochondria and chloroplasts are able to divide into two by a process known as fission. Now, this fission process is the way bacteria divide. The rest of the cell the cell itself divides by mitosis. They make that mitotic spindle, separate the chromosomes, and the whole thing. Bacterial fission, although it's complex, is a much simpler process. These guys divide that way. In fact, they can divide independently of the cell that they're in. Usually they divide when the cell divides, but that's not an absolute restriction. So we look at these things, biochemical similarities, the fact that they can divide by bacterial-like process, lipid composition, things like that. And when mitochondria and chloroplast DNA were first discovered, it really vindicated marvelous. And of course, this is the major theory for how these organelles originated in the past. In fact, I take a little look at that. Okay, and the theory that Margulis came up with, and like I say, she uses, is pretty much almost universally accepted now. So what's called the endosymbiotic theory. Now, endo means inside, and symbiosis, so we have two or more organisms that work together, cooperate for the benefit of both. An example would be a termite and the microbes living in the gut. The microbes have the capability of digesting cellulose, the termite can't do that. The termite has the capability of chewing up your house, the microbes can't do that. So everybody except you win. Okay, so it works for the mutual benefit of both. So endosymbiosis means one organism living inside another. And the idea is this. Mitochondria and chloroplasts have a lot of similarities between bacteria with bacteria because at one time their ancestors were real, honest-to-God bacteria. In fact, looking at the genetics, we can even place the closest living bacteria to mitochondria, looking at the genetic sequence, by this group of oxidative bacteria called alpha proteobacteria, which are very common and very diverse. The closest thing to chloroplasts, looking at the genetics, is certain types of cyano or blue-green bacteria, photosynthetic bacteria. So it looks like that's where their ancestors came from. But the whole idea behind the endosymbiotic theory is the whole idea is this. At some point, ancestral cells manage to get some different types of bacteria into them. And rather than digesting, which is what normally happens, in some cases the bacteria did not get consumed. Instead, they lived inside the cell, and it benefited both. Looking at genetic evidence again, it suggests that these processes probably occurred about 1.5 to 2 billion years ago, when we first started getting faint hints of eukaryotic, faint but controversial hints of eukaryotic cells. But the idea is this. An ancestral cell, and much of our genetics is quite similar, was no nucleus in these guys, an archaean. These are prokaryotes that look like bacteria but are very, very different. Some archaeans do not have cell walls, and they can kind of flow around a little bit and stuff. So it's possible that some of these guys could crawl around a little bit like an amoeba and maybe consume other types of cells. Well, somewhere down the way, we have, looking at, we'll take a bacteria. Bacteria gets into this ancestral cell here. Instead of getting consumed, it actually can do a big favor. Because this ancestral cell was probably anaerobic. In other words, it could not use oxygen in its metabolism. Might have even been poisoned by oxygen. But these oxidative bacteria that got in could. Not only would they remove the effects of toxic oxygen, this was an organism poisoned by oxygen, not only would they get rid of the oxygen, they'd also, by doing oxidative metabolism, generate a huge amount of ATP, far more than you could get by anaerobic process. So the guy comes in. All right, now ATP out. They produce all the ATP they want, pump some of the rest of it out. Now everybody's happy. Okay, for the bacteria, they're living in a much larger cell. They are safer. That cell may be able to crawl around and move towards food sources, away from nasty things. A lot of bacteria can't do that. So they got protection, a nice place to live. For the cell, they got the ability to use oxygen metabolism, tremendous energetic boost. So everybody's happy here. Now, the question is, now of course bacteria, they have cell walls and stuff. Okay, somewhere down the line, the cell wall disappeared. Probably good reason for it. Bacteria living inside that cell don't need a cell wall to begin with, and although cell wall is very porous, all it's going to do is slow down the transport of molecules, especially large molecules. If you're living inside that ancestral cell, you're better off getting rid of the cell wall. So mutations that prevent cell wall production were actually favored. You could do better without a cell wall than you could with. Now, if you're out in the open, that's a whole new ballgame. So they lose their cell wall. And then, over a period of time, they transferred their genetic functions to the nucleus, or future nucleus, of the host cell. Now, this whole thing is what we sometimes call horizontal or lateral gene transfer. And it's a major thing in evolution, passing genes from one cell or organism to the next. It's actually a major thing that leads and generates genetic variation. Now what can happen? Let's suppose you got a bunch of these symbiotic bacteria living inside this cell here. Some of them are going to die. When they die, most of the time the genetic material simply gets broken up. But sometimes pieces of that can actually get incorporated into the host cell's genome. So now, if you end up taking some of the bacterial genes and putting them in your own genome, you can produce those proteins. All you have to do is be able to transport them into the bacteria. And then, if the bacteria lose those genes, well, the nucleus, or the host cells provided that. So there's nothing against losing genes as long as there's a, quote, backup copy in the host cell's future nucleus. So after a while, you start losing more and more genes. More of those genes are getting transferred to the host cell. Mitochondria and chloroplasts still do have a few of their genes left, but less than, only about 3 to 5% of the total proteins they make. The rest of them are now in the nucleus, and the proteins get synthesized and transported in. Now, an interesting thing, although this sounds like a crazy idea, there's lots of evidence for it. And one of the most interesting things is there are a few species of protists that do not have mitochondria at all. Instead, they have real, honest-to-God, oxidative bacteria living in their cells. And these have real cell walls. They have your typical bacterial complement, about 2,000 genes or so. And you can grow them easily in culture. You can even separate one from the other. But if you separate one from the other, neither can be grown. They are absolutely dependent on each other, even though it seems like they have all their genes. Everybody's got all their genes and stuff. They are absolutely dependent on each other. They cannot grow without each other. So that's an interesting thing. In effect, we're seeing this endosymbiosis in a few cases happening again. Okay, well, that is a good place to stop. So you folks head down into the lab. We start